In this video, I'm going to discuss how to handle stop flow data collected with S159 CO2O2 analyzer and a gas switching system. In this case, we used a 4-channel gas switching system, and we used 3 of the 4 channels available. The S159 CO2O2 analyzer that was used had a CO2 analyzer in the range of 0 to 2,000 parts per million. And we used 2 small squash bug nymphs per chamber in channels 2 and 3, and channel 1 was our reference, of course. It had no chamber, just the solenoid valve assembly by itself. We used 2-minute dwell time, and measurements were done in mode 0. If you recall from the setup video, number of channels, dwell time, and mode have to be set up before you collect the data. Mode 0 is when you measure your reference at the beginning of each cycle. So you measure channel 1, followed by 2, 3, and back to channel 1. And we had the flow rate set at 140 mils a minute in this experiment. In the all 3 channels, it was quite close. And yeah, so we're looking here at page 1. Page 1 is where all of the raw data is displayed. As you recall, there are 3 pages, and we'll go through them one by one. The data has been collected as CO2, O2, the flow rate, the temperature, and the pressure at the oxygen sensor. The data is displayed in these graphs as well as the table. I'm going to stretch one of these graphs so we can look at it a little bit more clearly. As I mentioned, it's a good idea during setup, it's a good idea to run short experiments, trial experiments to begin with, to determine the best time for dwell time, the best setup, basically, the best flow rate for your experiment. So collecting few cycles of the data with your specimen to figure that out is a good idea, and that's what we've done here. And what you're looking for basically in your setup is you're looking for nice sharp CO2 peaks, nice sharp oxygen dips. You want to make sure that the dwell time period that you select is long enough to get the peak within that time period and reach a steady state low level. So basically flush the peak and then achieve that low steady state level before you switch to the next channel. Those are sort of the things you want to look for. You got to make sure that your CO2 peaks do not go above 2,000 parts per million, because that's the upper range of the CO2 analyzer. If the peaks are really high and reaching 2,000, you may want to reduce the dwell time, or possibly even look into open flow measurements if those peaks are quite high. So looking at this stretch graph, I'm just going to explain these flags or symbols that we have at the bottom here. The crosses represent the switch time from reference to the next channel. So basically, any measurements that are done before the red cross, those are the measurements done on channel 1. So you can see that here, and you can see that here as well. Red dots represent the switch from all the subsequent channels. So in this case, channel 2 is switched here and into channel 3. So data collected before that red dot and up to this red cross represents data collected from channel 2. And the orange triangle represents the switch from the last channel. Because we've selected only 3 channels for this experiment, this orange triangle represents the switch from channel 3, and therefore data collected between this orange triangle and the dot represents data collected in channel 3. So this is important to know and follow to make sure that you're flushing your chamber and that your peaks are not reaching the upper level of the CO2 analyzer, and the data that you've collected is going to be representative of what you want to do in your experiment. So that's our page 1. And as I mentioned, there are some user parameters that do not need to be set up before you collect your data. And I'll go through those now. If you go to the next page, which is page called differentials, this is where the software calculates the differences between the samples and the reference for CO2 and O2. So basically brings the CO2 and O2 data to the baseline of 0. So reference has been subtracted, and this is important because if you have any drift in your data, for example, oxygen, may drift a little bit. By subtracting the reference, you're getting rid of that drift. And I mentioned this before, the very first cycle, so this first cycle of data collected here should be ignored because the dwell time is not equal for all chambers and the system is being flushed out. So we don't look at the very first cycle. So I'm going to zoom into the data we have here and look at one cycle, the second cycle here, which is usable, and explain how to set these user parameters. So the parameters that we set are the event time, the oxygen delay, delay low, and delay high. The event time is a time period that is selected from the switch going backwards, and this period of time is usually about a quarter of the dwell time. So dwell time is 2 minutes, half a minute event time is sufficient, and it's the data that is selected and used mainly in the averaging of the reference levels and interpolation of the reference levels. So in this case, I've highlighted here about half a minute of that data, 0.415 of a minute, and you can see that it's nice and stable. So event time of 0.5 minutes is appropriate. The next parameter that needs to be set up is the O2 delay, and O2 delay is a parameter which basically lines up our CO2 with oxygen. The two are measured not exactly at the same time because the oxygen analyzer is after the CO2 analyzer, but obviously the changes are happening at the same time with the organism. So in order to do our calculation, for the software to perform the calculations correctly, we have to line these up. And sometimes the delay is very short, and sometimes it may be a bit longer depending on flow rate, depending on the volume of the chamber. But to do this, you go to Examine in the top menu, and you select that, and you'll get this line appear, the Examine line, and you can bring this to the peak CO2 and the dip in oxygen and see if they are totally lined up. Here you can see that they are off by probably 1 point. So you go to O2 delay and you increase that by 1, and you will now see that the two peaks have been lined up. So O2 delay of 1 is sufficient to bring these back on back into alignment. So that's the second parameter that needs to be set up. The third parameter is the delay low, and when the software calculates the VCO2 and VO2, it calculates it from the volume of the CO2 that's accumulated and volume of oxygen that's been depleted, and that volume is calculated from the area under these peaks or above the dip. So we have to tell the software where the peak starts and where does it end, and those parameters will depend on the flow rate through the system, the volume of the chamber, and activity of the animal. And to do this, you highlight first. We're going to select delay low. You highlight the data from the switch into that channel. So in this case, we're looking at data of channel 2. So the switch occurs here with the red cross, and you bring your you highlight the data up to the point where the CO2 and O2 start to increase, and you check in the corner here what that delta x is, and it's 0.332, and that is the number that you will enter under delay low. So we already have this entered here as 0.332. Click OK, and the black symbol represents where that starting point is, and it'll move into position once you've entered this number. The delay high is the point in time where the peak has already flushed through the chamber and a new sort of steady state has been, new steady state low, steady state has been reached. So that's basically the end of the peak, and that occurs right about here. Again, you look at your software here for delta x, it's 0.942. So that's what you would enter under delay high 0.942. I've already entered that number. So you click OK. The green symbol represents the end of that peak for integration. So the software now knows that the data between these 2 points is what needs to be integrated to calculate VO2 and VCO2. So once you've set these parameters, the software does all the calculations, and we can go to page 3, which is our summary page, where those data are displayed. So we have VCO2, VO2, and the respiratory quotient, which is the ratio of the two, displayed here. And the calculation and the formulas are explained, in this text box as well. And the actual numbers are going to be shown in the table below here. You have to remember that we have a single point for each dwell time for channel 2, which are the pink, and channel 3, which are the orange. So there is a single average point calculated for each one. And when you look at the data, I'm scrolling now down from the top, you will see, I just went over that, you will see here's the data for VCO2 and VO2 for channel 2. Okay, and then you go down further, and here is the data for channel 3, and these are the first couple of these points that are displayed in these graphs. So now you have two options. You can either take this data and copy and paste it into an Excel spreadsheet, which is actually what I've done right here. You can copy paste, for each point. You have to type in the titles yourself because they don't come across. Or your other option is you can export the whole data file by going to File, Export As, select CSV, and then name your file, exported file. I already had one saved here, so I'm just saving over that. And then you can go and open that exported file, which is right here, and look at it, and you will find that you will have here all of the data. Everything is brought across. So all of the raw data for each of the parameters, for each of the sensors. There's also going to be all the data that's used in control of the system, the switching. And right at the end is where you will find the data for the actual VCO2, VO2, and RQ for each channel. Channel 1 here, these are our reference numbers, which should be 0 or very close to 0. And then for channel 2 and for channel 3 are here. And they're again going to be individual points. And you have to basically scroll down and find each individual point. You have to do some manipulation of that Excel data. For example, go in and delete all these extra rows to create a single sort of compact summary table, and then you can take that data and do whatever statistical analysis you need to do, or graphing, or whatever else that needs to be done. So I think that's all there is really in terms of what to do with your stop flow data. As just a couple of final notes that I want to mention is, when collecting your experimental data, running your experiments, I mentioned this in setup files, setup videos. It's really important to save your data periodically. Excuse me, because LoggerPro does not automatically save data. So it's important to come in and periodically save your files. Make sure that if there's a computer meltdown or something happens, you don't lose your data. If you run your experiments over days, we in fact recommend that you restart every day with a new experimental file and save your data throughout the day to make sure that you don't have any lost data. So that's it. Thank you very much for listening.